Hello everyone and welcome to this new Gen webinar sponsored by Domain Therapeutics entitled Shedding New Light on GPCR Drug Discovery with a Novel Biosensor Technology. I'll be your host for today's webinar. I'm Jeff Bogaliskis, Technical Editor for Gen, which is going into its 40th year covering all aspects of the life sciences industry, from genome editing to bioprocessing to the ever important area of drug discovery, which we're going to hear more about in just a few moments. It would be next to impossible to speak about drug discovery and not include G-protein coupled receptors, as they constitute one of the largest classes of drug targets in the biopharmaceutical industry, as well as regulate virtually all known physiological processes. While much is known about the structure and function of these transmembrane receptors, there's still a significant number of human GPCRs that have yet to be successfully exploited as therapeutic targets. Investigators often noted that many current tools and screening methods are inadequate to address the various dimensions of GPCR signaling. In our webinar today, our presenters will tell us about an enhanced bystander bioluminescence resonance energy transfer-based biosensor technology called Biosensol, which addresses many of the current drug discovery bottlenecks that are limiting GPCR drug development endeavors. Now it's my pleasure to introduce our speakers for today's webinar. Dr. Arturo Mancini is a scientist at Domain Therapeutics, where he oversees various drug discovery activities exploiting the Biosensol GPCR pharmacology platform. Dr. Mancini will start off our presentation with a brief overview of GPCRs and introduce us to the Biosensol technology. Arturo will then turn his presentation over to his colleague, Dr. Maryam Samash, who is also a scientist at Domain Therapeutics and has extensive expertise in profiling GPCRs with the Biosensol platform. Dr. Samash will provide us with an extensive details of how the Biosensol technology can be utilized to study GPCR signaling. Finally, joining us for the Q&A session will be Dr. Laurent Sabag, who is head of biology for Domain Therapeutics, where he's currently leading discovery projects on GPCRs in immuno-oncology and CNS diseases. But before we begin, I want to remind the audience to stick around for the Q&A session that'll occur right after the presentation. All you need to do to ask a question is type your question into the Ask a Question tab on the right-hand side of your screen and hit submit. No question is too small. Send your question in at any time during the presentation and we'll try to get to as many of them as possible. Okay, now with all that said, I'm going to turn the mic over to Arturo and Maryam.
Hello everyone. To begin, I'd like to thank you all for your interest in our webinar. My name is Arturo Mancini and I'm a scientist at Domain Therapeutics North America. It's truly with great pleasure today that my colleague Maryam Samash and I present to you a novel, dynamic, breadth-based biosensor technology that allows for a multi-dimensional analysis of GPCR signaling and pharmacology. The presentation today consists of 4 main segments. To begin, I will provide a brief overview of G-protein coupled receptors and, more specifically, discuss current GPCR signaling paradigms as well as challenges and opportunities related to the therapeutic utilization of this receptor superfamily. This will be followed by an overview of the Biosensol GPCR biosensor technology. Maryam will then provide some specific examples of how the technology can be employed to study various aspects of GPCR signaling and pharmacology. She will then conclude with a brief presentation recap prior to opening the floor to questions. Before delving into the details of the Biosensol technology, and in order to fully appreciate the value of this technology, it is imperative to first review the many subtleties of GPCR signal transduction. G-protein coupled receptors represent the largest and most multifaceted family of human cell surface receptors, with over 800 members in the human proteome. These receptors are at the apex of essential signal transduction pathways, regulating numerous physiological functions, including vision, olfaction, and cell differentiation, to name a few. Consequently, the dysregulation of these receptors is frequently associated to disease, making them very important drug targets. Indeed, GPCRs represent the molecular targets of approximately 34% of all currently marketed drugs. GPCR-mediated signal transduction was originally believed to rely on linear signaling pathways, whereby a given GPCR selectively engages a signal heterotrimeric G protein family, defined by the nature of its G-alpha subunit. G-alpha proteins are encoded by 16 human genes and are divided into 4 major families, namely GS, GIO, GQ11, and G1213. Once activated, these G proteins each engage different downstream effectors that ultimately produce different signaling and thus biological outcomes. GPCR signaling is eventually terminated by a receptor C-terminal tail phosphorylation by kinases, including G protein receptor kinases, or GRKs, and subsequent receptor association of scaffolding proteins known as beta-arrestins. Beta-arrestins promote desensitization by sterically preventing G protein interaction through a process known as homologous desensitization, as shown by the black arrow here. They may also provoke receptor internalization and receptor ubiquitination by nucleating protein complexes involved in the latter two processes. It has become clear, however, that the classical model of G protein signaling that I just presented is an oversimplification of the full signaling potential of these receptors. Recent developments in GPCR biology and pharmacology have revealed remarkable complexity in how these receptors function. First, it is now clear that GPCR signaling can also occur via G protein independent mechanisms involving beta-arrestins. Once believed to solely mediate signal termination, these scaffolding proteins are now known to be critical signal transducers downstream of numerous GPCRs via their ability to nucleate signaling complexes, containing members of the MAP kinase and SRC kinase families. Furthermore, beta-arrestins can also be used by some GPCRs to activate receptor tyrosine kinase signaling, as has been shown for the epidermal growth factor receptor and the insulin-like growth factor receptor. Further, it is now evident that a given receptor can engage not only one, but multiple G protein families and beta-arrestins following ligand stimulation. What's more, GPCR signal transduction is not binary, but pluridimensional instead. That is, in response to a given ligand, any given GPCR can display different efficacies on the various signaling pathways associated to the receptor, resulting in the engagement of only a subset of receptor-linked pathways. The signaling output produced by a ligand is referred to as a signaling signature. So in this example, agonist 1's signaling signature is defined by activity on GQ11, GS, and beta-arrestin, and a lack of activity on all other receptor-linked pathways. Finally, and most importantly, different ligands can activate a given GPCR in different ways, with each ligand producing a unique signaling signature downstream of the same receptor. Thus, contrary to agonist 1 that engages GQ, GS, and beta-arrestin, agonist 2 engages G1213 and GIO pathways instead, likely producing a unique biological outcome relative to agonist 1. This concept, known as functional selectivity or ligand bias signaling, theoretically offers the opportunity to design molecules that preferentially activate signaling pathways important for therapeutic efficacy, while avoiding the activation of all other pathways associated with adverse events or drug tolerance. So at this point, I hope to have clearly conveyed that GPCR signaling is multifaceted and highly nuanced. Now, this complexity creates numerous therapeutic opportunities, but various important challenges as well. In fact, although 34% of all approved drugs act by modulating GPCR activity, it remains that only 16% of all GPCRs, or approximately 30% of the 365 so-called druggable GPCRs, have been successfully targeted. And this is illustrated in the dendrogram to the right, published by Alex Hauser from David Gloriam's group, in which each receptor lacking a colored dot is yet to be successfully targeted. Now, factors contributing to this limited targeting of GPCRs include the over 100 orphan GPCRs that lack tool compounds to aid in defining their functions, our incomplete knowledge of the complex signaling, biology, and pharmacology of many GPCRs, and the use of screening strategies largely based on classical GPCR signaling paradigms that fail to account for novel concepts in GPCR pharmacology. The end result is an overcrowding of the current GPCR target space. In fact, for the 130 GPCRs targeted by approved drugs, there are on average 10.3 approved drugs that use each receptor. Notably, aminergic receptors are targeted by 66% of approved anti-GPCR drugs, with certain receptors, as shown here, having anywhere between 20 to 50 drugs. Thus, we can identify novel chemical matter and novel drugs, but always targeting the same receptors. This not only results in a lack of novel drugs and targets to address unmet medical needs, but also makes it more challenging to differentiate these novel compounds from the numerous others already approved for the same target, thus increasing attrition risks. Clearly, there is a need to innovate the GPCR drug discovery and development pipeline by incorporating novel GPCR signaling and pharmacology paradigms into this process. This will necessitate the creation of novel technologies capable of assessing the various facets of GPCR signaling. Herein, we describe a novel, live cell-based assay that fulfills this technological need by allowing one to integrate emerging receptor pharmacology concepts, including allostery and functional selectivity, early in the drug discovery process. Application of this technology will help expand our understanding of the complexities underlying GPCR signal transduction and pharmacology, and in doing so, will help decode novel and difficult GPCR targets, expand the current GPCR target space, and unlock untapped therapeutic potential for this complex class of receptors, ultimately leading to the development of medicines to address unmet medical needs. So in the next few slides, I will provide you with a general overview of the Biosensol GPCR biosensor technology. The Biosensol platform includes an array of genetically encoded biosensors for monitoring both G protein dependent and beta-arrestin mediated signaling downstream of GPCRs. These biosensors are designed to measure receptor proximal responses that are engaged upon receptor stimulation, that is, G protein activation and beta-arrestin recruitment. Specifically, the sensors allow to detect the activation of 14 distinct heterochimeric G proteins as well as beta-arrestins 1 and 2, thus providing a multi-dimensional evaluation of signaling using the same assay system. Further, the platform permits the study of receptor and effector trafficking to early endosomes. In addition to proximal biosensors, the technology also includes sensors enabling the detection of distal effector activity and second messenger generation. The latter assays can be used to complement and/or confirm events observed with proximal biosensors. These distal sensors will not be discussed during today's presentation. So all in all, the Biosensol biosensor platform includes over 25 pathway and event-specific biosensors, each of which can be used for real-time kinetic analysis of signaling. Importantly, these sensors have been validated on over 100 wild-type receptors and over 2,000 naturally occurring variants, spanning various GPCR classes and including the enigmatic adhesion and olfactory GPCRs. So the biosensors at the heart of the technology are based on bioluminescence resonance energy transfer, or BRET, and more specifically, enhanced bystander BRET, or EB-BRET. BRET is a naturally occurring proximity-based phenomenon involving energy transfer between a bioluminescent donor, such as luciferase, represented here by an R, and a fluorescent acceptor, like green fluorescent protein, shown here as a G. The donor and acceptor are each fused to different proteins, represented by A and B in the illustration shown here, or to different parts of the same protein, all depending on if one is interested in detecting protein-protein interactions or conformational changes. In the presence of oxygen, luciferase catalyzes the oxidation of its substrate cilantrazine with the concomitant emission of light. When the luciferase is less than 10 nanometers away from the acceptor, through interaction of proteins A and B, for example, Forster resonance energy transfer occurs such that the acceptor protein absorbs the energy emitted by the luciferase substrate reaction and in turn generates a fluorescent signal at a specific wavelength. The final response is expressed as a BRET ratio, calculated by dividing the acceptor emission intensity by the donor emission intensity. Now, the EB-BRET approach used to generate biosensors in the Biosensol technology differs from conventional BRET in two main ways. First, EB-BRET uses luciferase and GFP from Rhinella reniformis as energy donor and acceptor, respectively. This naturally occurring donor-acceptor pair transfers energy more optimally than pairs used in standard BRET, resulting in an improved assay window and sensitivity. Second, The acceptor moiety in our EB-BRET system is actually not fused to a protein, but anchored to a specific cellular compartment instead, such as the plasma membrane or the endosomal membrane, for example. Turning our attention now to the assay principle underlying the biosensor platform. All biosensor assays are based on translocation of biosensor components to the plasma membrane and other endosomal compartments. For all G protein biosensors except GS, the subdomains of effector proteins that selectively interact with activated GTP-bound members of the GQ11, GIO, or G1213 family are fused to renylluciferase 2, RLUC2. These components are co-expressed with one of various G-alpha proteins and the receptor of interest. Upon receptor activation, the RLUC2 tagged effector translocates towards and interacts with the active GTP-bound G-alpha protein. This event brings RLUC2 fused to the effector protein in close physical proximity to the plasma membrane-anchored RGFP, leading to an increased BRET signal whose magnitude is proportional to that of the G protein activation efficiency. Exceptionally for GS, we're actually measuring dissociation of RLUC2 tagged GS from the plasma membrane upon its activation, therefore leading to a decrease in BRET signal upon GS engagement. The beta-arrestin assay is built on the same translocation principle, with beta-arrestin 1 and beta-arrestin 2 serving as the effector proteins that are recruited to the receptor upon activation. Now, there are a few points that I would like to highlight. First, the RGFP can also be anchored to various endosomal compartments, allowing one to assess trafficking and receptor activity from these intracellular signaling platforms. Second, anchoring of the BRET acceptor, the RGFP, to a specific compartment instead of fusing it to a protein provides a very significant advantage. That is, it allows to use untagged GPCRs and, in almost all cases, untagged G proteins. Third, all assays are miniaturizable for HTS purposes and are robust, with Z prime values ranging from 0.7 to 0.9. I'll now pass the floor to Marianne, who will provide some specific examples of how the technology can be employed to study different aspects of GPCR signaling and pharmacology.
Thank you, Arturo. My name is Mariam Samash, and I'm also a scientist at Domain Therapeutics, and I would like to also thank everyone for this opportunity. Through the next few slides, I will be presenting a few examples of applications in which the technology can be used. First application that I will be discussing is ligand signaling profiling and pharmacological mode of action studies. The first case study that I'm presenting here uses the niacin receptor GPR109A to illustrate an example of a typical profiling study for a given GPCR. GPR109A is a receptor that's been shown to display numerous beneficial effects once engaged, so anti-lipolytic, anti-inflammatory, and anti-atherogenic effects. The receptor can couple not only to G proteins but also to beta-arrestins. However, activation of beta-arrestins leads to adverse effects, more specifically inflammation at the level of the skin, and is a side effect that the majority of individuals taking niacin will experience. Through genetic model studies, it was shown that all the beneficial effects were mediated uniquely by the G protein signaling arm of the receptor signaling, while the side effects were mediated by the beta-arrestins arm. Based on these findings, tremendous efforts by pharma companies are aimed at developing compounds that would engage the G protein signaling arm of the receptor and avoid activation of the beta-arrestin signaling cascade. So we went back to what's been published, identified known binders, and profiled the whole series of ligands that were reported to determine whether the compounds that induced the toxic effect were indeed activating the beta-arrestin signaling arm of the receptor, and compounds that did not induce those effects were exclusively activating the G protein coupled pathways. Here, we are only showing 3 of the compounds as an example. Niacin, which is the natural ligand, is shown in blue, MK1903 is a small molecule that was reported to induce very little toxic effects and is shown here in red. Acifran is shown in green, and just like niacin, it induced all the toxic effects. So most of the sensors that were tested in this study are presented here, and what we can observe is that indeed, as predicted, the receptor engages the GI family of G proteins, so GI2, GOB, and GZ, in response to all the three ligands. Acifran had a slightly lower potency as compared to the two other ligands, but displayed the same efficacy. Now, when looking at the beta-arrestin pathway, we can clearly see that niacin and acifran, which both caused the toxic effect, were both able to engage beta-arrestin 1 and beta-arrestin 2 pathways. However, the MK1903 molecule that was designed to avoid these effects was much less active on the beta-arrestin pathways. So this was essentially a proof of concept that we can use this platform to profile compounds and differentiate them based on their signaling profiles and then relate these findings back to clinical observations. We can also interrogate a receptor and determine its signaling profile using biologics, such as antibodies. In this particular example, we demonstrate the activity of two polyclonal antibodies directed against a specific GPCR on two different pathways, the GQ and the beta-arrestin 2 pathway. The response induced by the natural ligand is shown in orange. The antagonist, shown in green here, completely reverses the response induced by an EC50 of the natural ligand on both GQ and beta-arrestin. What's interesting here is when we profile the antibodies in the allosteric antagonist mode, we saw that antibody 2 served as an antagonist on both pathways, whereas antibody 1 appears to display a partial allosteric modulator profile on the GQ pathway, but it was much more active in engaging the beta-arrestin pathway. So this is an example of a biased antibody. Allosteric modulators have been increasingly considered for the fine-tuning of ligand-based therapies. Allosteric modulators bind to an allosteric binding site that is distinct from the orthosteric binding site that binds the endogenous ligand, as well as classical orthosteric agonists. Allosteric modulators can induce conformation modification in the receptor that can lead to an enhanced or decreased affinity for orthosteric binding and/or efficacy, which may affect the receptor's biological activity. Here is an example of allosteric modulation. In this example, a dose-response curve of the human CB1 receptor-mediated GOB activation was generated with the Win55 agonist, and the dose-response curve is shown here in green. The human CB1 receptor is then treated with a negative allosteric modulator, PSN-CBAM1, and what you see here is a typical sheet plot analysis that shows the effect of increasing amounts of the negative allosteric modulator that results in a decrease in ligand-induced efficacy as well as a right-shifted potency of the reference ligand Win55. So this is a demonstration that this technology enables you to monitor this type of modulation. Another application is illustrated here, where we look at inverse agonists. Inverse agonists bind the same receptor as an agonist, but induce an opposite pharmacological response. They induce a reduction in the basal ligand-independent response. In our system, we work with transiently transfected cells, so we can control the amount of the receptor in our assays. When working with inverse agonists, we want to make sure that we have enough receptor expressed in our system to observe constitutive or ligand-independent activity. Here, the CB1 receptor was expressed at a level that was sufficient to have a high basal activity, so that when you add a documented inverse agonist, such as rimonabant here, you see that the inverse agonist decreases the signal below the basal level. So this is a clear demonstration that we can monitor inverse agonisms using the BioSense-All platform. The ability to detect constitutive activity of receptors is extremely important when working with inverse agonists. Inverse agonists can often be mistaken for antagonists, and this is due in large part to failure of screening assays to reliably detect constitutive activity. Being able to distinguish inverse agonists from antagonists is critical as they display fundamentally different mechanisms of pharmacological action. The rationale behind constitutive activity is that basal activity of any GPCR results from an equilibrium between inactive and active conformations of receptors. Receptor overexpression does not change this equilibrium, but increases the number of receptors in both conformations, hence reaching a threshold at which the active conformation can be detected owing to constitutive activation of a given signaling pathway. This threshold may be different depending on the signaling pathway to which the receptor is coupled. So when working under constitutive activity mode, we really need to look for inverse agonism with the right tools. Here is an example of an orphan receptor for which we monitored constitutive activity by measuring the Brett signal in the absence or presence of receptor expression in the system. On the left panel, we looked at the beta-arrestin 2 recruitment pathway, and a dose-response curve of an inverse agonist was generated in the presence or absence of the GPCR target. The data generated in the absence of receptor expression is shown in blue, and those obtained in presence of receptor expression are shown in red. In this example, the level of constitutive activity measured for the beta-arrestin 2 pathway is relatively small, as well as the effect of the inverse agonist. The green curve in the small upper panel is a control for agonist activity in the same condition. In contrast, when looking at the G13 pathway, we were able to detect a significant level of constitutive activity of the receptor and hence clearly distinguish the inverse agonist effect. These data show the importance to have multiple readouts to identify inverse agonists. The technology platform can also be used when comparing receptor orthologs. Most in vivo studies are conducted in rodents, mice models for example, and the rodent receptor does not necessarily behave the same way as the human receptor. So our system allows you to quickly compare the pharmacology of ortholog receptors as well. In this example, we compared the pharmacology of a target receptor that is coupled to GS. So it's a GS recoupled receptor, and if you look at the profile for GS activation, you can see that both receptor orthologs, the human versus the rat here, display essentially identical behavior. However, when you start looking at other pathways coupled to this receptor, so here GI2, GOB, G15, and beta-arrestin, you start seeing important differences in the way these orthologs signal. So having this kind of information early in drug development programs would allow you to improve study designs and identify a model in which the receptor pharmacology resembles to that in human, and further increase the chance of advancing therapeutics into the clinic. Here is a demonstration of how the platform can also be used to link clinical phenotype to a signaling profile. This is a collaborative study in which we profile various naturally occurring mutants of a variety of receptors in the human population. And with this study, we were able to show that there were not only mutations that displayed typical loss of function, but also mutations that induced bias in the coupling of the receptor. The three upper panels show the data obtained with receptor mutant 1, shown in red, compared to the dose-response curves generated with the wild-type receptor in black. As you can see, mutant 1 displayed a typical complete loss of function profile. Now, receptor mutant 2 is illustrated on the 3 lower panels in blue, and again compared to the wild type in black. On the GS pathway, you can see that the mutant had an almost identical profile as the wild type. However, it displayed a right-shifted potency when tested for G15 coupling and a complete shutdown of the beta-arrestin 2 recruitment pathway. Here is another example from the same study where we identified a mutant that induced a complete shutdown of a number of pathways. But in contrast, on the G13 pathway, not only this mutation induced a loss of basal activity of the receptor, but the ligand then displayed an agonist profile as compared to the inverse agonist profile initially observed on the constitutive active wild-type receptor in response to a ligand. The technology platform also allows for spatiotemporal analysis. A significant advantage that the platform can offer is the ability to perform real-time kinetics where the readout can be followed over time. So readouts for all the sensors can be done every few seconds for up to a few hours. This is an example in which we profiled the endothelin A receptor in real time using the different sensors. And what's interesting to observe here is that, for example, GRK2 and GRK5 overexpression enhance the kinetics of beta-arrestin 2 engagement. So the technology allows for the detection of bias that occurs at a certain time point post-ligand stimulation as well. Real-time kinetics can also be applied to the trafficking sensors. Here is an example in which we monitor GLP-1 receptor internalization and trafficking from the plasma membrane into the early endosome compartments. The receptor here is tagged with the luciferase at its C-terminus. In the assay shown in the left panel, a GFP is anchored at the plasma membrane, and breadth was measured upon receptor stimulation in real time. As a result, we see a time-dependent decrease of the breadth signal as the receptor disappears from the plasma membrane. In the right panel, the receptor is also tagged with the luciferase at the C-terminus, but this time the GFP is anchored at the early endosome compartment, and as opposite to what we see at the plasma membrane, we observe a time-dependent increase in the appearance of the receptor in the early endosome compartment. And this can also be done with beta-arrestin, because oftentimes beta-arrestin will co-internalize with the receptor, so you can also follow beta-arrestin internalization in a similar fashion as a surrogate for receptor internalization and desensitization. It has been demonstrated that endosomes are actively engaged in active signaling events. However, it has been particularly challenging to distinguish between the signaling events initially occurring at the plasma membrane from those at endosomal compartments. So, as shown in the previous slide, we have a similar GFP that is tagged to specifically localize at the endosomes rather than at the plasma membrane. So, in this configuration, we can do all the assays mentioned earlier at the plasma membrane, but this time looking at what's happening at the endosomes. So here is an example of GZ activation mediated by the delta opioid receptor measured either at the plasma membrane as illustrated in the two top panels or at the early endosome compartments, bottom panels. GZ activation is detected early on at the plasma membrane following receptor stimulation, and the signal is sustained for up to an hour. In contrast, maximum response in the endosome for GZ activation is detected almost an hour post-ligand stimulation. So these tools enable us to clearly distinguish between signaling events occurring at different cell compartments. A significant bottleneck in the discovery of new GPCR targeting drugs addressing unmet medical needs is the significant number of so-called orphan receptors, so receptors with no known ligands. The pharmacology and signaling attributes of these GPCRs remain inaccessible as endogenous or exogenous modulators have not been discovered yet. The biosensor technology offers two deorphanization strategies. The first strategy relies on the capacity of the technology to detect ligand-independent or constitutive effector coupling. As discussed earlier, constitutive activity is now widely accepted as an intrinsic feature of GPCRs, particularly when they are overexpressed in vitro expression systems. And this has also been observed in vivo studies as well. So, such constitutive activity upon overexpression of a GPCR can be used to identify the effector pathway involved in signaling by a given orphan receptor, thus mitigating the need for an agonist compound. In this approach, a receptor is overexpressed to various degrees and readouts are obtained on multiple pathways as shown here. Pathways showing receptor dose-response coupling, in this example GS, G15, can then be used for subsequent screening of compounds. This approach has been actually successfully applied in deorphanizing a receptor for one of our programs. The second strategy at deorphanization consists in combining the GZ and G15 sensors. We have recently completed analysis of G protein coupling for 100 GPCRs. This work revealed that 95% of these receptors activate either GZ or G15. Hence, the creation of a sensor that is capable of at once measuring activation of both pathways would provide a powerful and almost universal sensor that can be used for safety and systems pharmacology to detect the activation of nearly all GPCRs. Finally, the last application that we'll be presenting is EB-BRET imaging. The use of BRET has been largely limited to spectrometric measurements because of the low luminescence output that limits the spatio-temporal resolution required for high-quality imaging. Recent advances in detection devices with higher sensitivity and more efficient BRET probes, such as those offered by EB-BRET, now open new opportunities for the development of BRET-based imaging. Here, we show BRET-based imagery showing sensor recruitment to the plasma membrane upon receptor activation. HEC293 cells were co-transfected with a dopamine D2 receptor and the luciferase tagged effector for GI2 protein activation. BRET images were obtained in basal conditions and after stimulation with 1 micromolar of dopamine. In each image, BRET levels were expressed as a heat map color code, with the lowest being black and purple and the highest being red and white. After 200 seconds of stimulation, BRET signal is detected as shown here. This illustrates the ability of the technology to monitor time-dependent activation of different G proteins by EB-BRET imaging. The use of BRET has been largely limited to spectrometric measurements because of the low luminescence output that limits the spatio-temporal resolution required for high-quality imaging. Recent advances in detection devices with higher sensitivity and more efficient BRET probes, such as those offered by EB-BRET, now open new opportunities for the development of BRET-based imaging. Here, we show BRET-based imagery showing sensor recruitment to the plasma membrane upon receptor activation. HEC293 cells were co-transfected with a dopamine D2 receptor and the luciferase tagged effector for GI2 protein activation. PRET images were obtained in basal conditions and after stimulation with 1 micromolar of dopamine. In each image, PRET levels were expressed as a heat map color code, with the lowest being black and purple and the highest being red and white. After 200 seconds of stimulation, PRET signal is detected as shown here. This illustrates the ability of the technology to monitor time-dependent activation of different G proteins by EB-PRET imaging. So what I wanted to illustrate through these examples is the flexibility and variety of applications for which the platform can be used. So just to summarize, the technology offers a number of advantages. It offers the ability to work with unmodified receptors. So because most of the biosensors do not require tagging the receptor, it minimizes introducing possible artifacts into your studies. It also enables you, for example, to work with cell lines that express high amounts of receptors because of the great sensitivity of the assays. The technology offers the ability to evaluate all modes of pharmacological action, whether it is an agonist, inverse agonist, antagonist, or allosteric, and everything can be done in real-time kinetics. It is highly adaptable and can be used with small molecules, peptides, and biologics. It provides powerful tools for deorphanization. It is applicable to imaging, and it can be miniaturized for HTS campaigns. Finally, we would like to invite you all to view our manuscript describing the signaling profiling for 100 therapeutically relevant human GPCRs. It's available on the preprint server bioarchive, and the work presents an abundance of novel tools, approaches, and findings, and we invite everyone to view the data. I would like to thank you all very much for your attention, and we will be happy to take your questions.
Mariam, Arturo, wonderful presentations. Some really great insight into GPCR signaling and how the biosensor platform can really help overcome many of the challenges researchers are currently facing within this space. So we thank you for that. For the audience, our Q&A session is going to begin in just a few moments. Arturo, Mariam, and Laurent are all going to be there to answer any of the questions that you might have. So all you need to do is click the Ask a Question tab on the right-hand side of your screen, type in your question, and hit Submit. Okay, since we have a bunch of really great questions already lined up, let's get to the Q&A session. Bear with us for just a moment as we transition into the...
All right, everyone. Thanks for joining us for the Q&A session. We have a bunch of questions for our panel here, so let's get right to them. For the panel, this is going to be Mariam, Arturo, and Laurent. Guys, our first question comes in. One of our audience members would like to know, could you provide information that supports the translation of the data generated on recombinant systems to primary cells?
So yes, there are published studies in which some of the sensors have been used in primary cells, and that shows that our sensors were actually predictive of outcomes measured in physiologically relevant systems. In one of the published studies that I'm referring to, the signaling profile of the mu-opioid receptor was generated using different agonists, including bias agonists, and this was first studied in HEK cells, where bias was actually confirmed. And in parallel, the same ligands were tested in isolated and cultured neurons from transgenic mice expressing a tagged form of the receptor. So looking at readouts like receptor internalization and trafficking, and the data showed a nice correlation between the two different experimental systems, supporting in vivo translatability of bias signaling of the mu-opioid drugs. Another study also on the mu-opioid receptor was published, and in which the authors described the signaling diversity of mu-opioid receptor ligands characterized using the sensors. Then unsupervised clustering was performed, which allows for the classification and association of different clusters to the frequency of appearance of the side effects that were known and reported. And one last study that I wanted to mention is a project in which the signaling profile of allosteric agonist of the receptor GPR40 was conducted in human pancreatic islets. And the readout was measuring glucose-induced insulin secretion. So these are examples that support the translation of the data generated on HEC cells recombinant systems to primary cells.
Thanks, Mariam. I'd also like to add a few points to that. So basically, the data in HEC293 cells will tell you which couplings are possible. So that is, in response to a given ligand, can my receptor adopt a conformation that allows it to couple to a given effector? Now, obviously, how well the data in the HEC293 system translate to a more physiologically or pathophysiologically relevant system depends on various factors, for example, including receptor expression levels in the tissue or the cell of interest, relative expression levels of various receptor isoforms for those receptors that have different isoforms, where we see sometimes that various isoforms can have very different signaling outputs. The same for ligand levels and for receptors that have multiple ligands, where different ligands can each produce their each unique signaling profile. And of course, the relative levels of expression of the various effectors, so the G proteins, the arrestins, GRKs as well should not be ignored. So the field is moving forward very quickly, I think, to connect the in vitro and in vivo sides of the equation with the development of large-scale protein expression data sets across various cells, across various tissues, and in producing broad protein interaction maps, for example. But I think a critical question is how can the tools that were presented today be best used to derive translatable data? And I think the answer lies in using signaling signature data to cluster ligands based on their signaling profiles, and then testing one or two exemplar ligands from each cluster in vivo as quickly as possible to ultimately help determine which type of signaling profile or bias, if you will, produces the desired outcome in vivo.
All right, thanks, guys. So for our next question, one of our audience members would like to know, or they asked, it says, in your assays, would the functional selectivity be affected by the different cell types?
So I think this brings out a very significant advantage in our system. So cell type or system bias is more likely to be observed when measuring events further downstream in the receptor signaling cascade, such as second messenger levels or distal effector activity. And this is because the further downstream along a signaling cascade that a response is measured, the greater the number of intermediary regulatory factors or regulatory nodes that may modulate the downstream response. And the factors in these nodes include various kinases, various scaffolding proteins. Now, what's important here is that the expression of these intermediary factors may vary from one cell type to another cell type, and thus potentially producing cell type specific imprints on the downstream readout and ultimately producing system bias. So as was presented today, our biosensors were really measuring receptor proximal events that are the first responses engaged following receptor stimulation. That is G protein activation and beta-arrestin recruitment. So we believe that since we're measuring events at the apex of a signaling cascade, we're much less susceptible to cell type specific or system bias. Further, we conducted basically an exercise in which we, for a few receptors, profiled different ligands in your classical cell line, so HeLa cells, COS, CHO, U2OS. And with those receptors and ligands that we tested, we observed that ligand rank order potency and efficacy were conserved across the different cell backgrounds.
All right, thank you. Next question. Audience member would like to know, how sensitive is this technology?
So this technology is highly sensitive, allowing to measure the activity of endogenously expressed receptors. And we've actually even worked with endogenous G proteins, and we are able to pick a signal from that. And also, as mentioned in the seminar, the sensitivity of the assays allow for the detection of constitutive activity, which is advantageous for deophylizing receptors, or to identify inverse agonists in the sense that it's sensitive enough to pick constitutive activity.
Yeah, and also, I mean, we work with different types of ligands, different types of molecules. So from small molecules to peptides to antibodies, biologics. And so, you know, we and with especially with the biologics, we're looking at sub nanomolar potencies, which we can very easily pick up with our system. Further, I can say that we're definitely capable of picking up all partial activity, and for some of the supposedly biased mu-opioid receptor ligands, we were actually to pick up very, very small, for example, beta-arrestin recruitment, where we're looking at maybe 10% relative to a reference non-biased ligand. So the system is definitely from a pharmacological perspective extremely sensitive as well.
Okay, and for our next question, the audience member says that the system looks great, but since G is anchored to the membrane, how does that allow to measure changes in molecules that are not recruited to the membrane, like PKC, for example?
Yeah, so it's important to note that the PKC sensor is actually built on a different principle. So we're not looking at translocation of PKC per se. We're actually looking at PKC activity, and these sensors are really not the main focus of today's presentation. But for that particular PKC sensor, we basically consist of a surrogate that, when phosphorylated by PKC, will impart a change in breath signal. So really, it's not this particular sensor is not based on a translocation principle as was presented today by Mariam for all of the other sensors, but based on a slightly different principle. And just to add, this same principle applies to a cyclic AMP biosensor that we also have in our pipeline of assays.
All right. Thanks, guys. Next question. Do your biosensors bind to the extracellular domain and or to transmembrane domains of GPCRs?
So the biosensors per se are interacting. They're built to interact with the G alpha proteins. The beta-arrestin itself actually interacts with the cytosolic side of the receptor. So that's again another advantage of the system, in that when the G alpha protein undergoes a GDP to GTP exchange, so when it basically becomes catalytically active, the sensor will directly interact with and only interact with the active form of the alpha protein. So it's actually not built to directly interact with the receptor, but built to directly interact with the active alpha protein.
All right, thanks. Next question. The audience member says, great talk, and would like to know, what are the approximate absolute values of Brett ratios with biosensor technology, as standard Brett assays often tend to have very small values of Brett ratios that leads to variability. So it's a two-part question. Next part, it says, using real-time kinetics, how can one distinguish whether beta-arrestin recruitment is for receptor internalization or for activating G protein independent pathways? I'd be happy to go over the second part again if you want to start with the first part.
Yeah, so to answer the first part, basically, raw Brett values will change from instrument to instrument, and at least, on our instruments, we're looking at raw Brett values of 0.2 going all the way to above 1 for certain assays. So the full change is quite considerable with the enhanced part of the bystander, right, that we've basically done. That's thanks to the GFP and the R loop that's being used in our system. So it's quite a significant assay window. With regards to whether beta-arrestin recruitment, being able to differentiate whether beta-arrestin recruitment is related to internalization, sensitization, or coupling, signaling. So basically, I'll have two answers for that. Number one is that in order to tease out whether arrestin recruitment is linked to internalization, we can also in parallel run internalization assays with our system. So that would confirm if beta-arrestin recruitment is somehow involved with the internalization of the receptor. Secondly, there's a sort of a second flavor of biosensors, which again were not presented here today, but which look more at the conformation of the beta-arrestin versus just its simple recruitment. And we know that, for example, based on principle or in a so-called phosphorylation barcode hypothesis, where different phospho residues on the receptor C-terminal tail will stabilize different beta-arrestin conformations, each of which can lead to different functional outcomes. So we can actually, with our sensors, the conformational beta-arrestin biosensors, we can possibly use these to distinguish whether a molecule is inducing beta-arrestin recruitment to internalize a receptor, or if there are additional functions such as activation of ERK and AKT pathways. So it's really to answer that second question, it's really we have multiple tools that can be applied, I think, in a combinatorial fashion to answer those questions.
Okay, thanks, guys, for that very long question, but you guys hit it great. Next question. Audience member asks, can you comment on the critical experimental conditions for accurate and sensitive measurements?
So prior to all assays, receptor expression is optimized. This is really a key variable in the assay. But other than that, the assays are extremely robust because we work with a very streamlined and standardized protocol. We make sure that we work with very consistent transfection conditions to work with the exact same conditions. Yeah.
Okay, thank you. Another question. What is your opinion on GPCRs in which the receptor coupling molecules are unknown? For example, angiotensin type 2 receptor.
Yeah, so I think Mariam presented a strategy based on constitutive receptor activity or pre-coupling, where we would titrate the different amounts of receptor into our system, and at a certain point hope to have enough receptor in an active conformation that will be able to basically pick out a pathway that can couple to the receptor. So this is one strategy that we may use to basically characterize or expose the coupling partners for receptors where no coupling molecules are known.
All right, and it looks like we have time for one last question. Regarding GPCR activation assays, what are the effectors transfected to translocate near the GTP anchor?
So we work with effector proteins that recognize the alpha-subunits in an alpha-subunit specific manner. So we have effectors for the GI family, another for GQ, for example. And these have been published in the bioRxiv platform that we mentioned earlier. So we really invite everyone to go and really take a look into the validation and everything related to that.
All right, great. Thanks, guys. And with that, we've come to the end of our webinar. So I'd like to remind everyone that the webinar will be archived on the GEN website for up to a year at genengnews.com. So if you missed any parts of it, you can watch it again. Feel free to forward the link to any of your friends and colleagues, which we always recommend. I'd like to thank Mariam, Arturo, and Laurent again for their informative presentations and Q&A session. And I'd like to thank the audience for their attention and very thoughtful questions. And a very special thanks to Domain Therapeutics for sponsoring this webinar. Hopefully we'll see you again in another Gen webinar in the near future. Goodbye for now. Everyone stay safe and healthy.
Thank you, Jeff.